Good afternoon, my name is Gert Pietersen and I will be telling you about Grapevine Yellows phytoplasma detection methods. The detection methods I will be covering will include the sample preparation process, followed by primers and how they bind to DNA in the detection assays. I will talk about PCR amplification and its endpoint or real-time detection. I will then speak to you about Sanger sequencing, which is used for the genetic analysis of strains. And then lastly, I will briefly cover how LAMP detection methods work. DNA is extracted from plant or insect samples. This can be done by one of typically two formats, either by making use of organic solvents followed by precipitation in a manual and quite laborious process, but that yields good quality DNA, such as illustrated in the left-hand picture. Or it can be done by kits which make use of spin columns, and these are quite expensive but very time saving. Primers are short segments of single-stranded DNA molecules which bind to the target site of detection assays, such as in PCR and in LAMP. They bind in a complementary fashion to single-stranded DNA, which is when the native double-stranded DNA is exposed to high temperatures, it denatures, separating into two separate strands, allowing for primers to bind. The complementary bases combine in such a way that A's will always bind to T's, C's will always bind to G's, and vice versa. DNA polymerase chain reaction makes use of a purified DNA template, which contains a mix of host DNA as well as potentially the pathogen of interest, in our case phytoplasmas. As previously mentioned, they make use of primers which bind to the complementary start and end regions of the target sequence. PCR then makes use of a cycle of 3 steps which repeats, resulting in the exponential amplification of the target DNA of interest. This consists of denaturing at high temperatures, which splits the double-stranded DNA molecule into its single strands, annealing, which is a lower temperature at which primers bind to the complementary target sites, followed by elongation, wherein the thermostable Taq enzyme, DNA polymerase, extends the DNA target site. PCR reactions can be altered in various ways to increase their sensitivity or specificity. One such approach is by making use of nested PCR, wherein the DNA product of one PCR reaction is used in a subsequent downstream PCR reaction. In this case, 2 sets of primers are used. In the first round of PCR, outer primers cover a larger region of the target DNA, followed by inner primers which bind within the target region for the second round of PCR. This is a great method to increase the sensitivity of one's assay and also leads to a successively higher specificity in downstream reactions. This, however, is quite sensitive to contamination by carryover or from adjacent samples. The amplification reaction can be tracked by typically one of 2 ways. The older, more traditional way is an endpoint analysis, whereby the final DNA product, called an amplicon, is loaded onto an agarose gel, which is then subject to gel electrophoresis and fluorescence by UV light. The product can then be visualized by UV fluorescence and its size determined as compared to a molecular weight DNA marker, which is loaded with the gel. The other method is called real-time or quantitative PCR, which, as the name suggests, continuously monitors the reaction as it proceeds. This graph is the typical output you would expect from this kind of analysis, and it on the one axis displays the number of cycles of the PCR which have completed versus the fluorescence which is observed inside the reaction, which is as a result of either a hydrolysis probe being degraded due to the PCR reaction, or by intercalating dyes which only bind and fluoresce to double-stranded DNA, which accumulates in the reaction as it proceeds. Sanger sequencing is a further analysis that can be conducted on a PCR product. It makes use of chain termination, which is not an exponential amplification, but rather a linear process whereby DNA products of varying lengths are created, which is used in capillary electrophoresis, by which data is generated that looks like this chromatogram that you can see on the slide. This sequence data can be analyzed then to determine genetic variation, from which strains and species can be elucidated. This approach was made use of in the TROPICSAFE work package 1 surveys, which were conducted on phytoplasma in fynbos and looking for alternative vectors, as published in the reference at the bottom of the slide. The last topic that I would like to cover with you for today is loop-mediated isothermal amplification, or LAMP. This is also a DNA amplification method which makes use of 4 to 6 primers. However, in this case, the primers bind in a more complex fashion and such that they form structures which fold back on themselves, called dumbbell structures. These form repeated units called concatenates of progressively larger fragments, and very quick rates of exponential increase in DNA in this kind of reaction, which is also monitored digitally in real time as a standard practice. This kind of reaction is carried out by an enzyme called the BST, DNA polymerase, which is less prone to inhibition. The reaction is also carried out at a constant temperature of about 65 degrees, reducing the complexity of the reaction. This makes it ideal for point-of-care testing, and in fact an LAMP assay like this has been designed for phytoplasma detection by our partners in Slovenia at the NIB, National Institute of Biology for Slovenia.